Today we'll be talking about the cellular transport. First off, I'll be talking about diffusion. Diffusion is the movement of molecules, either gas or fluid, that moves from high concentration to low concentration. In the diagram, you will see the molecules naturally moving to the area of higher concentration, which is the left, to lower concentration, which is the right. Next, osmosis is the diffusion of water that moves from a region of high water concentration to low water concentration. In the diagram, you can see that the movement of the water is heading outside of the cell because outside of the cell contains more sugar. Three types of solutions in osmosis. One of these are called the isotonic solution. The isotonic solution is when the amount of solutes and water molecules are equal and the water moves equally in and out of the cell. As a result, the cell would either get shriveled or lysed. A hypertonic solution is where water moves out of the cell because there are more solutes outside of the cell. As a matter of fact, the cell would shrivel. Last but not least, the hypotonic solution happens when a cell gains more water than is outside because there are more solutes in the cell. As a result, the cell would swell up and eventually pop, if it's an animal cell. Now things are a bit different for plant cells. If once a plant cell gains water, it doesn't pop easily like the animal cells. It would not pop because of the strong cell wall, but only the cell membrane would swell up. This can be also called the trigger pressure. For the hypertonic solution, where the water would leave the cell, the animal cell would easily shrivel. However, the plant cell does not shrivel because of its strong cell wall. Instead, the cell membrane would shrink. This process is called plasmolyzed. Now, because we know diffusion, we'll also get to know what facilitated diffusion is. So, what is it? Facilitated diffusion is a type of passive transport that uses the help of proteins to carry big molecules into the cell. Passive transports are like the facilitated diffusion and diffusion. It doesn't use ATP because it is going with the concentration gradient range, and it goes from high concentration to low concentration. On the other hand, the active transport uses ATP because it goes against the concentration gradient range and also moves from low concentration to high concentration. In summary, the difference between the passive transport, active transport, and the facilitated diffusion is that passive transport does not need the use of ATP because it is flowing, or should I say, going with the concentration gradient range and moving from high concentration to low concentration, which does not need energy, while the active transport needs ATP because it's going against the concentration gradient and also moving from low concentration to high concentration. So basically it is rising. And last but not least, facilitated diffusion is different to all because it cannot squeeze through the membrane just the way it is. It needs the help of proteins. Endocytosis' main job is to basically take in substances into the cell. There are two types of endocytosis called the phagocytosis and the pinocytosis. Endocytosis is a type of endocytosis, but is responsible for taking in bigger particles. In the diagram, you will see the process of phagocytosis, taking in the food particles and storing them in the vacuoles. Also, pinocytosis is a type of endocytosis that usually takes in smaller substances and water. Exocytosis is pushing substances out of the cell, such as the removal of waste. In the diagram, you will see the vesicle transporting the molecules and then getting released by the use of ATP. Finally, sodium and potassium can be also seen as an active transport example. By the help of ATP, they pump out 3 sodium ions per 2 potassium ions.